Okay, so what we're going to look at today is hemoglobin, and we'll do kind of an intro to dissociation curves. So first of all, you need to know some stuff about red blood cells, and in particular hemoglobin. Hemoglobin is a protein found in really high concentrations within red blood cells, and its job is to effectively carry oxygen to or from the lungs to respiring tissues. And so you can see here we've got molecules of oxygen that are joining onto the hemoglobin on the red blood cells. And here at this end, you've got the oxygen molecules coming off. Now we give these, or give the name, they've got specific names. When it joins on, we call that association. And when it comes off, we call that dissociation. And so in this case, we've got the oxygen associating with the hemoglobin. And then you've at this end got the oxygen dissociating. So what we're going to look at is how the oxygen joins on, and then some of the processes behind how it comes off or dissociates. So, hemoglobin itself comprises, it's a quaternary structure, and the reason why it's quaternary, it has more than one polypeptide chain. It has 2 alpha subunits and 2 beta subunits, and for each hemoglobin, each of those subunits, there's a heme group. This heme is prosthetic, which means it can basically be removed. And it's surrounded by a Globin chain, and hence why it's called hemoglobin. The Globin chain is the sequence of amino acids, which is obviously a polypeptide chain, and it is obviously unique. So you can see that it's folded, and so it's a globular protein. When it forms oxyhemoglobin, this is when an oxygen molecule joins to the heme group, and so that's why it's referred to as that, oxyhemoglobin. Okay, so for every hemoglobin molecule, you're able to adhere 4 oxygen molecules, because obviously 4 heme groups, as you can see there. And at the centre of each heme group is an ion. Ion, as you can see there, Fe2+. So, if we took a look at the kind of the structural order of proteins, in this case, the primary structure, so the sequence of amino acids, then the secondary structure, that would be forming an alpha helix. Now you can see here they've deliberately messed up the sequence here. So this is technically the tertiary structure, this diagram here, because it shows it being folded into a unique 3D shape. The tertiary and the quaternary structure here, again in the wrong place, should be quaternary. That's because it comprises of more than one polypeptide chain. So in this case, sequence of amino acids for the 4 polypeptide chains is your primary structure. That will then coil into helixes, or alpha helixes in this case, and then due to the specific sequence of amino acids, it will fold in a very unique way. And then the quaternary structure is meaning it comprises of 4 different polypeptides, and so therefore it can carry 4 oxygen molecules. So, just to clarify on these terms, associate means to join with the oxygen. So when you form oxyhemoglobin, that's because the oxygen has associated with the red blood cell and the hemoglobin. And then when it dissociates, that's when the oxygen comes off it. So you can think of dissociation as letting go of the oxygen, and you can think of association as them joining on. So the way in which to remember this and where this happens, then you're going to associate the oxygen onto the hemoglobin. You're going to do that at the gas exchange section, so basically in the lungs. So gas exchange and grab. You're going to disassociate at respiring tissues. So in this case, where you get respiring tissue, so muscles, any of the organs, you're going to release it. And so you can think about these 2 different areas. It's going to associate in the lung and disassociate at the tissues, specifically the respiring tissues. So one of the things that you have to kind of understand here is a process called positive cooperativity. Now what that means is that effectively every time an oxygen associates with hemoglobin, that makes it easier for the next one to bind on. So if I show you a diagram illustrating this point. So this shows positive cooperativity. Now in this case, this is without any oxygen being joined. And you can see here the green bits represent the heme group, and then you've got your Globin chains in this blue and red here. So every time an oxygen molecule joins on, because there are obviously four polypeptide chains here, it makes it easier for the next subsequent one to bind, because you can see here this Globin chain has become less tight and easier to bind to. Now that's true up to the third one. So every time an oxygen molecule binds, it becomes easier for the next one to bind on. So we call that positive cooperativity. Now, that isn't true for the final one, and I'll come to explain about that in a second. But basically, every time you add an oxygen to a hemoglobin molecule, so for the first three, it gets easier for the next oxygen to bind. And so you can see here, this is the difference between with having none joined and having several joined. In this case, you can see this is in the relaxed phase, so the oxygen has a much easier time of binding, whereas in the tense phase it's much less likely to bind. And so we call positive cooperativity when an oxygen atom binds to one of the hemoglobin's four binding sites, the affinity of the three remaining available binding sites increases. So it just means it's more likely that the oxygen is going to bind to the heme group. Now, that's the case for the first three. So you can see here that if we've got this graph, and this graph you're going to come across quite a lot of time. So this shows the partial pressure of oxygen on this x-axis, and on the y-axis you've got the fractional saturation, basically the saturation level. So this means this should be, I mean technically this is fractional saturation, but this is normally expressed in percentage. So 1 is 100%. Now what you should be able to make out here is that at very low concentrations of oxygen, so at low partial pressures of oxygen, you can see that the level of saturation of the hemoglobin is quite low, and that it gets progressively, it's got this S-shaped curve, so it gets progressively easier for the oxygens to bind, and that's down to that positive cooperativity. Now the only exception to that is when it gets to 3. So from 3 to 4, the reason why the level of cooperativity goes down is because, in effect, if you look at when there's basically, if there were 3 joined here, the likelihood of the next oxygen molecule being able to bind to that spare one is less likely. So the oxygen molecules are effectively moving around and hopefully colliding into the right area. And because once you've got 3 filled up, the likelihood of filling the 4th up becomes lower. That's why there isn't this positive cooperativity, and in this case it's negative from the third to the 4th, sorry. So what we see there is it becomes much more difficult for it to become fully saturated from 3 to 4 for that reason, but you always get this S-shaped curve. So you can see here at lower partial pressures there isn't a great deal of change, but when you do start to increase the partial pressure of oxygen, you can start to see greater saturation. And at the top, the partial pressure doesn't change the saturation that much at this point. So you get this S-shaped curve rather than it being a straight line curve. Now, this means, so this going back to the positive cooperativity, as soon as the first oxygen binds, the first to the 4th binds, distorts it, making it more relaxed. This makes it easier for the 2nd and 3rd to join, and it basically makes it more difficult on the last one because there's only now one binding site for the oxygen to possibly join to, and therefore becomes more difficult. So in terms of changing its affinity for oxygen, you can see that if you've got at the gas exchange surface, this is at the lungs, if you've got high concentration of oxygen, you've got a low concentration of CO2, the affinity for the hemoglobin is really, really high. And so this means that you're going to attach loads of oxygen to each hemoglobin. Now at respiring tissues, you can see that the oxygen concentration is low, CO2 is high, the affinity is much lower, and so the oxygen gets released. So this attachment means association, and this one means dissociation. And this is really useful because the hemoglobin picks up the oxygen at the lungs because there's a lot of oxygen concentration, and it means when it gets to the respiring tissues there's not as much oxygen, so it disassociates. So by disassociating, it means that it comes off in the blood, and then it means it can cross the membrane barrier on the capillaries and the membrane wall of whichever muscles or whichever tissues it's reached, so it can get into the cells. So it's a good thing, because without this change, you wouldn't be able to disassociate. The oxygen remains on the red blood cell and you wouldn't be able to get it off. So there are two major types of hemoglobin for this. You've got some types which have a really high affinity for oxygen, and you've got other types that have a lower affinity. So an example of this might be in an organism that lives at high altitude. Now, at high altitude, you've got much thinner air, so the partial pressure for oxygen is much lower. And what basically that means is, it means that the hemoglobin will be able to take on a lot more oxygen at lower partial pressures, which is really useful for organisms that live up mountains. Now, for organisms that maybe don't live up mountains but have a high respiration rate or metabolic rate, they need to disassociate the oxygen quickly. So where maybe the level of oxygen in the atmosphere isn't a problem, they might have this version. So in this case, if the organism has got a really high metabolic rate and the level of oxygen in the atmosphere isn't a major problem, they're going to have that type or that subvariant of the hemoglobin, because it's going to disassociate quicker. And what determines the different subtypes is basically the amino acid sequence. So if you change the amino acid sequence, you change the primary structure, you change the secondary and the tertiary structure, you therefore make a type of hemoglobin that can associate at lower or at higher partial pressures. Okay, hopefully that's been of help.